What's really new this year in the management of myelofibrosis is in the United States is the FDA approval of Fedratinib with a very broad indication of previously treated or previously untreated patients with intermediate and high-risk myelofibrosis. And Moshe, you did some of the very initial work with Fedratinib. Why don't you talk to us about the road to FDA approval for that drug?
So yes, we work with Fedratinib for more than 10 years. It used to be a Target-gen drug, so it changed to Sanofi, it changed to Celgene.
Changed to Bristol.
The company changed to Bristol, okay. So we really participated with it from the phase 1. And the experience, the overall overarching statement would be not a bad drug, it's a good drug. The second statement I would make, it's not as good a symptom controller as ruxolitinib is. I think it doesn't have the same percentage. It's more like 30%. And maybe one of the reasons is that it perhaps has more side effects. Especially in the first month or two of treatment, it has more, mainly gastrointestinal side effects, like nausea, vomiting, diarrhea, and that may counter the positive symptom effect. The third point is that I, at least personally, have been impressed with its effect on splenomegaly, which exceeds that of ruxolitinib. And one good example is we have done a limited phase 2 study testing 3-dose level. To the extent that I recall, it was 300, 400, 500 milligrams, and found that 40% of the patients that were quote-unquote resistant to ruxolitinib responded with a spleen response almost equal to the same numbers that you get frontline with ruxolitinib. So that was an impressive number. It was to an extent confirmed in the larger studies that were done with Fedratinib. And I give credit to Celgene for now going back, reanalyzing the studies and being extremely conservative with the analysis, but they still have a nice 30% response rate in this population of patients. Many of them were totally resistant to ruxolitinib. So it has probably a superior effect as far as spleen control. Katriana Jamieson and I published a paper where the argument was that it also reverses fibrosis in a substantial number of patients, whereas that control of fibrosis was restricted to a rare and small number of patients with ruxolitinib. Now, with a word of caution, this has to be proven prospectively in an organized study. I wouldn't like to overread into it, but I'd like to see a study. I do have patients who have improvement in fibrosis, no question about it, but this is anecdotal. This is not a solid basis for argument. Two more arguments, scientific arguments. One argument is the claim to fame that it's not a JAK1 inhibitor, it's a JAK2 inhibitor, so maybe it has less immunosuppressive effects, maybe. The second argument is more like my argument. The Fedratinib is a multi-kinase inhibitor. It's not no drug in the world is a pure kinase inhibitor that inhibits only one kinase, but this one happens to inhibit many. And it's very possible that many of its effects are not a function of just inhibition of the JAK2 kinase, but also other kinases, and that has to be studied a little further. Generally speaking, it adds to the armamentarium of drugs. It's a JAK2 plus drug, and it clearly has a role not only in patients that are resistant to ruxolitinib, also perhaps in patients who, to start with, have very large spleens. It may be a superior drug in this patient. So we will identify eventually the niche for each one of these drugs.
You know, when I look at as we remember the COMFORT-1 study, it did not require a JAK2 mutation. About 75% had a JAK2 mutation. Yeah. And so we couldn't sort out the benefit of the drug in JAK2 mutated or unmutated. And I'm not a statistician, but when I look at the waterfall plots, it seemed to me that there were more patients with a JAK2 mutation who had splenic size reduction than JAK negative patients. But that was just my...
But was there not a preponderance of JAK2 patients to go into those trials as well?
Right. Yes, there was.
You know,
It was
even though it
75
didn't have to
It
be.
was, but it didn't look the same.
The
in
prevailing claim is that they're all the same.
Yes.
That as far as response, it doesn't matter if it's calreticulin, JAK2, or MIPL, they all respond equally, although prognostically they are not exactly the same, where MIPL is considered a worse disease, a more serious disease. To what extent this is confirmed, or we just go with these statements as it goes along without actually confirmation, I don't know.
Along those lines of being potentially good in patients with lower blood counts, we do have one, Claire Harrison's presenting here at ASH, that there's a specific analysis of Fedratinib with patients between 50,000 and 100,000, where they're still seeing
You're
meaningful,
correct, that's another point.
yeah,
Yes.
symptom and spleen response. So it does bring in the question of, well, I'm curious, would you think about frontline starting with Fedratinib rather than ruxolitinib in a
Well,
patient with lower platelets?
Robin, you bring up a very important point. Is it less myelosuppressive as far as the platelet effect and the anemia than ruxolitinib? It has to be looked at carefully.
So what are the downsides of starting with fedratinib in terms of toxicities? Mary Frances, I mean, what do you think about that?
My experience is going back to the trials, and the number of patients on the whole tolerated it very well, but I gather that a lot of them will have a lot of GI toxicity, particularly in the first month or two, and in fact there's advice now provided to anticipate this problem and give them antiemetics and advise them on antidiarrheals up front.
John, he didn't talk about Wernicke's encephalopathy.
Yeah, maybe because there's not much to talk about. So, there was a concern early on that led actually to the halting of the clinical development when it was under the control of Sanofi-Aventis due to 8 cases of Wernicke's encephalopathy. And when the dust settled and those were really evaluated in a rigorous fashion by an independent committee, it was probably more like 1 case. And there really hasn't been a lot of literature that would support. I've personally checked thiamine B1 levels in my patients with myelofibrosis. I don't see thiamine deficiency. I've not seen Wernicke's. I do pay attention to it. There is a black box warning now. So the advice is to check thiamine levels before and then I think every 3 months while on therapy. The reality, it's probably cheaper just to put someone on a multivitamin and send them on their way. But it wouldn't persuade me not to prescribe the drug.